All right, so we're talking about ketone bodies. Now, ketone bodies they act as an alternative fuel for cells. Now, what exactly are ketone bodies? They are water-soluble molecules which are made in liver mitochondria from acetyl-CoA during periods when the body is using a lot of fat. So, the times when body is oxidizing a lot of fat, obviously, where does beta oxidation of fatty acids happen? It happens in mitochondria and particularly in the mitochondria of liver. So, there, these water-soluble molecules are made, which are ketone bodies. So basically, from beta oxidation, you get acetyl-CoA, and this acetyl-CoA it gets converted to ketone bodies. There are three ketone bodies that should be on your fingertips: acetoacetate, beta hydroxybutyrate, also known as 3-hydroxybutyrate, and acetone, which is non-metabolizable, breathe it out, fruity smell. So you have these three things: acetoacetate, beta hydroxybutyrate, and acetone. Now, moving forward, why does liver make the ketone bodies? As I've already told you, when you're fasting, you're in a starving state, or you're exercising heavily, or you have uncontrolled diabetes, liver is going to receive a huge amount of fatty acids from adipose tissue. Now, in adipose tissue, in your lactating memory tissue, and also in your liver, all of them collectively synthesize fatty acid. So when the body is exercising or is in a starving state, fasting state, or has uncontrollable diabetes, all of this fatty acid it goes to liver and there it undergoes beta oxidation where it will be broken down into acetyl CoA. But at this point, the TCA cycle is also slowed down. Why? Because during fasting already the body doesn't have glucose, so it's already in a glucose deprived state. Therefore, all the oxaloacetate that is in the part of TCA cycle that is being used for gluconeogenesis. So TCA won't occur because TCA does what? It hydrolyzes glucose in glycolysis and then it goes into TCA in order to make energy. But right now already the body is in an energy deprived or glucose deprived state, so you're not going to break down more glucose. Instead we are going to go for gluconeogenesis. So all of that oxaloacetate in the TCA is going to gluconeogenesis. So acetyl CoA it cannot enter TCA cycle, therefore this acetyl CoA is going to accumulate. What does the liver do when liver sees कि, oh, there's so much acetyl CoA and it's not going to TCA cycle? So let me do one thing, let me convert it into ketone bodies. Now, ketone body synthesis in the liver is called ketogenesis. What happens in ketogenesis is that again during fasting, adipose tissues release fatty acids, liver takes them up, beta oxidation, and this massive acetyl CoA production because this acetyl CoA isn't going into TCA cycle. Instead, this because oxaloacetate is being used for gluconeogenesis. So what about this acetyl CoA? Acetyl CoA will be converted to ketone bodies. Now, high acetyl CoA does two things. First thing first, it inhibits pyruvate dehydrogenase. So pyruvic acid oxidation doesn't happen, and acetyl CoA pyruvate is not made from. Pyruvate doesn't get converted to acetyl CoA. So acetyl CoA is not made from your pyruvate. Stops acetyl CoA from being made from glucose, or you can say pyruvate. Now, the other thing this does, massive acetyl amount, does one more thing, which is that it activates pyruvate carboxylase. Now, pyruvate carboxylase is an enzyme that supports gluconeogenesis, and therefore, what happens is that the acetyl CoA there will be more oxaloacetate to support gluconeogenesis. Yeah, so one thing it does is it inhibits pyruvate dehydrogenase. The other thing it does is pyruvate carboxylase, where the pyruvate is getting converted to oxaloacetate. So there'll be more oxaloacetate to support gluconeogenesis. Since TCA cycle is still slow, the liver diverts acetyl CoA into ketone synthesis. Acetyl CoA gets converted first to acetoacetate, which is one of the ketone bodies. Um, Acetoacetate is then reduced to beta hydroxybutyrate or spontaneously decarboxylated to acetone. So first, via HMG CoA synthase and HMG CoA lyase, aceto acetyl CoA is converted to acetoacetate, and then it's reduced to beta hydroxybutyrate or it's decarboxylated to acetone. Ketone body used by peripheral tissues, that's called ketolysis. Now, when this ketone body is going to go to the peripheral tissue, their ketolysis will happen. So, where is ketogenesis taking place? In liver. What about ketolysis? It's taking place in the peripheral tissues. Who uses ketone bodies? Skeletal muscles, cardiac muscle, kidney, intestinal mucosa, and brain. Only during prolonged fasting, brain will be using ketone body. Now, why do they use ketones? Because ketone bodies spare glucose. So they spare glucose. This protects glucose for the RBCs, which depend only on glucose. Now, I told you that even your brain, it solely tries really hard to depend on glucose, but in extreme cases, ketone bodies will go to the brain, and obviously, brain needs energy. And what about RBCs? Now, RBCs, they cannot use anything other than glucose. So glucose is left for your spared for your RBCs. Now, how tissues use ketone bodies? Beta hydroxybutyrate, it's converted to acetoacetate. Beta hydroxybutyrate is again converted back to acetoacetate. Acetoacetate plus CoA taken from succinyl CoA, acetoacetyl CoA, split into two acetyl CoA, enter TCA for energy. So basically, ketone bodies, once they go back to the tissue, they're going to again get converted to acetyl CoA, and that acetyl CoA there it will enter the TCA cycle. What's the important point? That the liver cannot use ketone bodies. It lacks the enzyme thiophorase, succinyl CoA, acetoacetate CoA transferase. Because from succinyl CoA, you have to transfer the CoA, right, to your acetoacetate. So that enzyme thiophorase is lacking. So it produces ketones, but it never uses them. So there's one organ that doesn't use ketones, that is your liver. And another structure in the body, RBCs. RBCs, they never use ketones. They are solely dependent on glucose. And then excessive ketone body production in diabetes mellitus. Now, in type 1 diabetes, no insulin is there, massive lipolysis. Obviously, if there won't be any insulin, there'll be a lot of lipolysis. That is fatty acid degradation pathway. Flood of fatty acids to liver, extreme beta oxidation, huge amounts of acetyl CoA. TCA cycle cannot keep up. Liver, because TCA, how much of that acetyl CoA will go into TCA? TCA cannot keep up because obviously at the end of the day, it's a fast procedure, but still it's relatively slow compared to the amount and rate at which the lipolysis is happening and beta oxidation is happening. Liver overproduces ketone bodies, blood becomes acidic, ketoacidosis, and excess ketones spill into urine, and that's ketonuria. And also, that person has a fruity breath, acetone. Now, quick summary box. When ketones are made, fasting, starvation, prolonged exercise, low carb diet, and controlled type 1 diabetes. Why? Excess acetyl CoA plus low oxaloacetate, and even though oxaloacetate is low, they can't enter TCA cycle. And why is oxaloacetate low? Because it is going for gluconeogenesis, right? I told you that. First thing first, acetyl CoA is excess and low oxaloacetate because your this enzyme was not functional. Which enzyme was I exactly talking about? Right here they mentioned activates pyruvate carboxylase. This is activated and this is inhibited. And yeah, especially for what do? They provide an alternate fuel, especially for brain, muscles, and kidneys. Who cannot use them? Liver and RBC. So this is such a good um, summary.